Hi, my name is Bohdana Doskaliuk, and I'm an associate professor at the Ivano-Frankivsk National Medical University. Today, we will be talking with you about the modern nanomaterials, their classification, structure, and properties. Also, we will be talking about the methods of nanomaterial synthesis for biomedical approaches. So I would like to disclose that I do not have any conflict of interests. And let's talk about the nanotechnologies. What is their relevance? Nanotechnologies are already transforming reality, opening new perspectives in disease treatment, diagnostics, and the development of novel pharmaceutical forms. What are the nanomaterials? They are materials which particle size is in between 1 and 100 nanometers. This grants them unique properties compared to the conventional materials. These characteristics are widely used in medicine and biology. What should be the key properties of nanomaterials? First of all, high surface area to volume ratio. It can increase the reactivity and binding capacity of nanocarriers. It is critical for catalytic and sensing and drug loading applications. Nanomaterials also should have biocompatibility and biodegradability. It is essential for biomedical use, especially in drug delivery, imaging, and tissue engineering. Nanocarriers also should possess physicochemical stability. Often, nanomaterials also present very suitable optical and magnetic properties. They can enable advanced applications in diagnostics like fluorescence imaging or photothermal therapy, and also targeted therapy. If we would like to classify the nanomaterials, there will be a few classifications. First, we can do it according to the chemical composition. All nanomaterials then can be divided into main three groups: organic, inorganic, and the hybrid. To organic, we can include liposomes or nanogels or dendrimers. To inorganic, it's metallic nanoparticles, metal oxides, carbon nanotubes. Hybrid, it's the combination of organic and inorganic components. By the structure, we can differentiate the nanomaterials into the 0-dimensional, 1-dimensional, 2-dimensional, and 3-dimensional. 0-dimensional are the quantum dots. 1-dimensional is nanotubes, for example. To the 2-dimensional, we can include the graphene or nanosheets, and the 3-dimensional are, for example, the nanosponges. Also, we can classify the nanoparticles by their chemical composition. We have the group of metallic nanoparticles that are widely used due to the excellent physicochemical properties. Usually, they can be seen in the drug delivery, biosensing, imaging, or as even like antimicrobial agents. We have the group of semiconductor nanoparticles. Mostly, these are the quantum dots. They have the ability to be highly responsive to the intermediate conductivity. The third group are the nanoshells. They are the core shell structures composed of the core material encapsulated by the shell. There are two main types: oxide nanoshells and metal nanoshells. Usually, they are used in targeted therapy and diagnostics. So let's talk a little bit more about dimensions of the nanoparticles. So what does it mean actually that the nanoparticle is 0-dimensional? It means that all dimensions are within the nanoscale. For example, quantum dots. They are widely used in electronics due to the strong quantum confinement effects, enhancing optical and transport properties. The 1-dimensional is the nanoparticle, whose 2 dimensions are within the nanoscale, but one dimension exceeds 100 nanometers. The example can be the nanowires or the nanotubes. The two-dimensional are characterized with one dimension remained within the nanoscale, but with the two others which are larger. The example here are the nanosheets and nanocoatings. And last but not least, the three-dimensional nanoparticles are those where none of the external dimensions are within the nanoscale, but the internal features exist at the nanoscale. The example can be fullerenes and dendrimers. They are composed of lower dimensional building blocks with broad applications in advanced nanotechnology. By the structure and morphology of the nanomaterials, we can divide them to two big groups: the crystalline structures and amorphous structures. Crystalline structures defined by highly ordered atomic or molecular arrangements. They are characterized by regular geometric patterns, often leading to distinctive optical, magnetic, or electrical properties. They exhibit predictable mechanical strength, thermal conductivity, and electronic characteristics. The example can be the gold nanoparticles. Amorphous structures lack long-range atomic order. Their atoms randomly distributed through the nanocarrier. Generally, they are possessing less uniform properties compared to the crystalline counterparts. Advantages for the certain biomedical applications, they can be due to the increased solubility and bioavailability. The example of the amorphous structures can be silica nanoparticles. We can also differentiate the two groups of the core shell structures and mesoporous structures. Core shell structures composed of the nanoparticle core encapsulated by a shell material, allowing multifunctionality. The core typically imparts unique optical or magnetic properties, while the shell provides biocompatibility, stability, or targeting capability. Widely applied in controlled drug delivery, bioimaging, and catalysis. Example: gold core silica shell nanoparticles are popular for photothermal therapy due to the stability and biocompatibility. Mesoporous structures, on the other hand, characterized by pores ranging from 2 to 50 nanometers. They possess exceptionally high surface area, enabling effective drug loading and controlled release. Commonly used as carriers for anti-cancer drugs and therapeutic agents, ensuring sustained delivery and reduced toxicity. The example can be mesoporous silica nanoparticle. On this slide, you can see actually the comparison of the core shell and the mesoporous structure. From the left side, you can see the core shell structure with the core that consists of the gold, and the coating, the shell, is of mesoporous silica. On the right, you can see the TM picture of the mesoporous structure of the silica, and you can observe it on the different scales of magnification. So what is the role of morphology in the biological interactions between the nanocarriers, nanomaterials, and our body? It is critical as the influencer of interactions. For example, it can affect cellular uptake efficiency, biodistribution and clearance rate, toxicity, and immune response. For example, spherical nanoparticles typically exhibit high cellular internalization. Rod-like or tubular nanoparticles possesses prolonged circulation time due to the evasion of immune clearance. Rod-shaped nanoparticles may also cause greater mechanical stress to cells compared to the spherical particles. On this slide, we can observe the different ways how the nanoparticle can interact with the cell. It can be internalized and induce the signaling by also being inside of the vesicle. They can just attach to the receptor without even entering the cell. The nanoparticle can even be internalized, and it can accept the vesicle and can even interact with the various organelles of the cell. At the bottom of the picture, at the picture part C, we can even observe the multiple transferrin-coated gold nanoparticles that were internalized by the cells of the HeLa line, and we can see them inside of the intracellular vesicles. On this slide, you have two QR codes which you can use to get more familiar with the process of interactions between the nanoparticles and the cells. Let's talk a little bit more in detail about the structural features of nanomaterials. So we were talking already about the shape matters, but how it matters. So the spherical particles will be commonly used in the drug delivery due to their features. Rod-like or tubular structures, they will be ideal for scaffolding or directional transport. Cubic forms will be often used in catalysis and photonic applications. The particle size is also very important. We will talk about it a little bit later. Interesting feature is also the surface charge or zeta potential. It determines the colloidal stability and interaction with biological membranes. Positively charged particles may enhance uptake by negatively charged cell surfaces, but can also increase cytotoxicity, unfortunately. So what about the shape? Spherical gold nanoparticles, approximately 50 nanometers, demonstrated higher uptake in the HeLa cell lines compared to the rod-shaped nanoparticles of equivalent volume. Spheres are also more efficiently wrapped by the plasma membrane, making them easier for the cell to internalize. Rods may experience higher energy barriers for internalization due to their elongated shape. The different nanoparticles that have different shape can even interact with the organelles differently. Rods tend to accumulate near the nucleuses and have prolonged intracellular retention. Spheres are more evenly distributed and may be cleared more rapidly. Cytotoxicity may vary with shape. While both shapes can be functionalized similarly, rods may trigger different levels of reactive oxygen species and interfere with cytoskeletal organization due to their geometry. So the different nanomaterials with the different shapes will have the different applications. Spheres will be best for the fast uptake, drug delivery, and clearance. Rods or discs potentially useful for long-term retention, imaging, or mechanical interaction with the cells. If we will be deciding what size of the nanoparticle we should choose, we need to remember that the size will define the uptake efficiency. The optimal uptake efficiency occurs at approximately 30-50 nanometers core diameter. It is large enough to engage multiple receptors, but small enough to be internalized efficiently. If the nanoparticle is less than 200 nanometers, it will be internalized via clathrin-mediated endocytosis. If the size is already between 200 and 500 nanometers, the endocytosis may shift to the caveolin-dependent, or even phagocytic pathway. Extremely small nanoparticles, which are less than 10 nanometers, may diffuse or even adhere to membranes before the uptake. The size of the nanoparticle can even modulate the subcellular localization. Nanoparticles of 2 to 10 nanometers can enter the cytoplasm or even nucleuses, whereas larger ones, like more than 50 nanometers, typically will remain in endosomes or lysosomes. Here we can observe the two illustrations about the uptake of nanoparticles by the mesenchymal stem cell from the left and by the red blood cells from the right. And we can observe the differences in the uptake according to the size of the nanoparticles. So from the left, we see that the most active uptake and is exactly at the level of 8 nanometers in comparison to 100 or 3.3 nanometers. Let's talk about the surface charge, or zeta potential. It is the potential difference between the dispersion medium and the stationary layer of fluid attached to the nanoparticle surface. When nanoparticles are dispersed in a liquid, ions in the solution are attracted to the nanoparticle surface, creating an electrical double layer. Zeta potential is measured at the slipping plane, which is the boundary between the stationary layer of fluid attached to the nanoparticle and the bulk fluid. Optimal zeta potential is either plus or minus 30 millivolts. Generally enhances stability and biocompatibility. Zeta potential is a key indicator of the stability of nanoparticles in suspensions. A high zeta potential, either positive or negative, indicates strong repulsion between particles, leading to greater stability and preventing aggregation. Low zeta potential suggests weak electrostatic repulsion, which can lead to particle aggregation and sedimentation. Zeta potential plays a role in how nanoparticles interact with biological systems, influencing their biocompatibility and potential for drug delivery applications. On this slide, you can see how zeta potential can be different in accordance to the different level of the pH in our organism. Also, we can divide the nanoparticles into the hydrophilic or hydrophobic. So, first let's talk about the formation of the protein corona. What does it mean? Nanoparticles, which are entering the bloodstream, get coated by proteins. That is actually protein corona. Surface hydrophobicity drives stronger protein absorption, and in contrast to hydrophilic coatings, repel protein binding creates a stealth effect. Fewer absorbed proteins means reduced immune recognition. Solubility and stability will also differ between hydrophilic and hydrophobic nanoparticles. Hydrophilic surfaces disperse well in water, preventing aggregation. Hydrophobic surfaces tend to clump together to avoid water. In physiological fluids, this leads to poor colloidal stability. Hydrophobic nanoparticles may aggregate in blood, forming larger particles. Aggregation can hinder circulation and even cause capillary blockages in extreme cases. The biodistribution, it also will differ. Surface chemistry alters where nanoparticles travel in the body. Hydrophilic stealth nanoparticles absorb fewer opsonines, so they evade phagocytes and circulate longer. They can more readily reach target sites before clearance. In contrast, hydrophobic nanoparticles with heavy protein corona are quickly opsonized and sized by the mononuclear phagocyte system. These nanoparticles often accumulate in clearance organs rather than the intended target. So on this slide, we will briefly summarize the advantages and risks both of hydrophilic and hydrophobic nanoparticles. So the advantages of the hydrophilic nanocarriers lies in the high water compatibility. It means better solubility and stability in biological fluids, which will reduce aggregation. Minimal protein absorption confers longer blood circulation, longer half-life. For example, pegylated nanoparticles resist protein folding and avoid immune clearance. From the other side, hydrophilic surfaces reduce interactions with cell membranes. This leads to poor internalization unless active targeting is added. Long-term or repeated exposure to pegylated nanoparticle can induce antibodies. This leads to the accelerated blood clearance phenomenon, which will reduce nanoparticle efficiency and alter distribution. Hydrophobic nanoparticles. The hydrophobic surfaces can penetrate the cell membranes more readily and improve cellular uptake of payloads. The lipid-like character interacts favorably with the phospholipid bilayer, enhancing direct membrane translocation or endocytosis. Moreover, hydrophobic nanoparticle matrices excel at carrying hydrophobic drugs, allowing encapsulation and controlled release of otherwise water-soluble therapeutics. This makes them effective for delivering lipophilic drugs into cells. Poor water solubility means that hydrophobic nanoparticles often aggregate in blood, which can trigger unintended immune responses or even physical vessel blockage in severe cases. Adsorbed plasma proteins rapidly tag hydrophobic nanoparticles for clearance by macrophages, leading to short circulation time and accumulation in organs like liver and spleen, which will decrease the targeting effect and also may cause off-target effects. On this slide, we have the summary of the different features of hydrophilic and hydrophobic nanoparticles that you can use for a nice sum up. We cannot forget also about the ability of biofunctionalization of the nanocarriers. Yeah, so we can modify the surface, and the modification can be covalent or non-covalent. The covalent modifications involve forming strong chemical bonds between functional molecules and the nanoparticle surface. Such covalent attachments are stable and typically persist under biological conditions. Covalent coupling often allows multi-step functionalization and can incorporate multiple functions without the risk of the ligand coming off easily. On the other hand, non-covalent modifications use reversible absorption of molecules onto the surface through weak interactions such as electrostatic attraction, van der Waals forces, hydrophobic interactions, etc. Non-covalent attachment is relatively simple and preserves the molecule structure and activity. However, because these bonds are weak, the attached molecules can detach if the environmental conditions change. This means non-covalent coatings are useful for loading drugs that should be released at the target site. There is also the phenomenon of ligand-directed targeting. Attaching specific biological ligands to the nanoparticle surface enables active targeting of the nanoparticle to certain cells or tissues. The ligand on the nanoparticle recognizes and binds to a matching receptor that is abundant on the target cell. This ligand-directed approach greatly improves the specificity and nanoparticles delivery. As the result, there is less interaction with normal cells, which reduces off-target effects and side effects. In drug delivery, such targeted nanoparticles can deliver higher drug doses to the tumor while sparing healthy tissue, thereby enhancing the treatment efficacy and minimizing systemic toxicity. On this slide, you can observe the two different types of ligand-directed targeting, active and passive. Basically, passive targeting relies on the enhanced permeability and retention effect, or EPR effect. The nanoparticles can accumulate in tumor tissue and release the encapsulated drug to the cells by the diffusion. Usually, the tumor tissues will have more leakage in their blood vessels and less active lymphatic tissue. So, due to that, the nanoparticles can accumulate exactly in that particular area, even without using the specific ligands. We have mentioned today a few times the process of pegylation, or PEG. So what does it mean? Pegylation refers to attaching polyethylene glycol polymers to the nanoparticle surface. PEG is a flexible hydrophilic polymer that forms a water-rich cloud around the particle, effectively camouflaging it from the immune system. This stealth coating prevents protein in the bloodstream from opsonizing the nanoparticle and reduces recognition by immune cells. The result is that pegylated nanoparticle circulated longer in the blood because they are not rapidly cleared by the liver or spleen. In addition, pegylation lowers the immunogenicity. The particles after this process will be less likely to provoke an immune response. Moreover, if we are talking about functionalization, we can also provide the antibody conjugation. Attaching antibodies to the nanoparticle equips them with a highly specific targeting mechanism. Antibodies naturally recognize unique proteins, antigens. By conjugating antibodies to a nanoparticle, researchers create a guided missile that seeks out cells carrying exactly this target. The antibody functionalized nanoparticles can precisely bind to disease markers and accumulate at the disease site. This precision targeting leads to enhanced delivery to the target and reduces uptake by normal tissues. We can perform also the peptide functionalization. Short peptides can also serve as targeting ligands when attached to the nanoparticles. One prominent example is the RGD peptide, which binds to integrin receptors that are commonly overexpressed on tumor cells and on the blood vessels that feed tumors. Coating a nanoparticle with RGD or the other targeting peptides directs it to those integrin-rich sites. This leads to selective accumulation of the nanoparticles and improves the uptake by the target cells. Because peptides are smaller and can be engineered or modified easily, they provide a flexible option for the targeting. For example, the RGD-decorated drug carriers have been shown to deliver drugs more efficiently to cancer cells while reducing adverse effects on normal cells. So how are nanomaterials designed and produced to achieve such unique properties? If we will try to classify all of the synthesis methods, so we can divide all of them into two big groups, top-down and bottom-up. In a top-down synthesis, one starts with the bulk material and breaks it down into nanoscale particles. In contrast, bottom-up approaches assemble nanoparticles from atomic or molecular components, as in the chemical precipitation or atomic deposition. The top-down physical methods often produce very pure products, since minimal chemical additives are used. They can be scaled to bulk quantities. However, they tend to require intense energy input or specialized equipment. The bottom-up chemical or biological methods usually occur under milder conditions and allow fine-tuning of particle size or shape by adjusting reaction parameters like precursor concentration, pH, or temperature. Chemical syntheses are typically cost-effective and can be scaled up in liquid reactors, but they involve chemical reagents, some of which might be even toxic. Green biological synthesis aims to avoid hazardous chemical entirely, making the process more eco-friendly. So let's start discussion of physical methods of synthesis. There are three main types of physical synthesis. There are the laser ablation, mechanical milling, physical vapor deposition. Laser ablation is a high-power pulsed laser beam which is focused onto a solid target. Often it is a metal. The aim is to vaporize a tiny region of it. The ejected plum of atoms rapidly cools and condenses into nanoscale particles. This technique can be done in vacuum, gas, or liquid environment. Laser ablation can produce ultra-pure nanoparticles because the only material involved is the target itself. Another advantage is the ability to generate material types that might be difficult to synthesize chemically, including composites, simply by ablating a mixed target. However, drawbacks include the need for expensive laser equipment and high energy consumption. It can also be challenging to tightly control particle size distribution. Often, particles have clean surfaces but may agglomerate if no stabilizing agents were present. Mechanical milling, or ball milling. Mechanical milling is a top-down method in which bulk material, powder or solid, is pulverized into nanoparticles by grinding with hard balls or media in a rotating mill. High-energy ball milling repeatedly fractures and cold welds the material, gradually refining it down to nanoscale. The advantages of this method are its simplicity and scalability. It also does not require chemical reagents, so the product can be quite pure. Limitations include limited control over the particle size and shape. The process often yields a broad size distribution from tens to hundreds of nanometers. Physical vapor deposition. It refers to various vacuum-based methods where a material is vaporized and then condenses as the thin film of nanoparticles on a substrate. Examples include sputtering, bombarding a target with energetic ions to eject atoms, and thermal evaporation, which includes heating a source until it vaporizes. PVD can produce nanoparticles, coating, or even free nanoparticles. These methods offer excellent control over composition and cleanliness. Moreover, physical vapor deposition can be used to make well-defined nanostructured films. The downsides are the PVD equipment is very expensive and it requires technical expertise. Okay, so on this slide you can observe the pros and cons of the physical methods of the synthesis. They are summarized here for you. Let's talk about the chemical methods. Chemical methods are bottom-up approaches that involve building nanoparticles from atoms or molecules through the chemical reactions. Typically, the person can start with the metal atoms in the precursor's form and induce their assembly into the nanoparticles via processes such as precipitation, reduction, or thermal decomposition. Chemical syntheses often occur in solution and allow for a high degree of control by tuning reaction parameters. Key chemical synthesis techniques include sol-gel processing, chemical reduction of metal salts, and hydrothermal synthesis. Let's start from sol-gel synthesis. So, from this slide on, if you will see any QR code, it means that you can use it to have the access to some video on the YouTube that can help you to understand and more visually interact with the processes of the synthesis of the nanoparticles. So, sol-gel synthesis is the versatile bottom-up chemical technique to produce metal oxide nanoparticles using metal alkoxide precursors. The process of this synthesis will include the hydrolysis and condensation that will lead to the colloidal sol. Then we will have the formation of the semi-solid gel, and at least there will be the process of drying and calcination that will lead to formation of crystalline nanoparticles. What can be the advantages of this method? First of all, it's high control over size, shape, and composition. It operates at mild temperatures. Sol-gel synthesis enables organic and inorganic hybrid nanoparticles and produces monodisperse biocompatible particles. Chemical reduction of metal salts is a widely used bottom-up method to synthesize metallic nanoparticles by reducing metal salts in solution. Metal ions like gold or silver are reduced by agents like sodium borohydrate or sodium citrate, forming stable colloidal nanoparticles. The advantages are those that this method is simple, scalable, cost-effective, it has fine control over size and monodispersity, it is compatible with surface functionalization, although it also has some drawbacks. It may leave residual byproducts like borates or surfactants, and as the result, it requires purification for biomedical use. Hydrothermal synthesis. It's a method where reactants in water are sealed in a high-pressure autoclave and heated to 180–250 degrees Celsius. This enables crystal nucleation and growth at elevated pressure and temperature, but without boiling. The pros of this method is that it produces highly crystalline, uniform nanoparticles. It enables the phase control. It has low contamination risk, and it is water-based, so it is eco-friendly. The limitation are those that the hydrothermal synthesis requires high-pressure equipment like autoclaves, reaction times may be pretty long, and it is sensitive to solubility and supersaturation control. Biological, biogenic, or green synthesis of nanoparticle. It employs the living organisms or biomolecules to produce nanoparticles. In these bottom-up approaches, the reducing power and chelating ability of biological extracts are harnessed to convert metal ions into nanoparticles. This often occurs in water at mild conditions, even at room temperature, and near neutral pH, making it an environmentally benign process. Additionally, the biological molecules that perform the synthesis tend to remain on the nanoparticle surface as a capping layer. There are two main groups of this green synthesis. First one, microbial synthesis. Certain microorganisms have the natural ability to reduce metal ions and precipitate them as nanoparticles either inside or outside their cells. This can be a defense mechanism or simply a byproduct of metabolism. For instance, the bacterium Bacillus subtilis and the fungus Fusarium oxysporum have both been reported to synthesize silver nanoparticles when exposed to silver salt solutions. Typically, the microbes secrete enzymes or metabolites that reduce the metal ions. In bacteria, NADH-dependent enzymes can reduce ions, and proteins can nucleate and stabilize the growing nanoparticles. In fungi, extracellular enzymes and rich cocktail of metabolites can perform a similar role. On this page, you see the summarization of the pros and cons of the microbial synthesis of nanoparticles. The next one is the plant or photosynthesis of nanoparticles. This method is very eco-friendly. And in this method, the extract of the plant is mixed with a metal salt solution. The phytochemicals serve as the both reducing agents and stabilizers, causing metal ions to be reduced and then keeping the resultant nanoparticle. The reaction is often indicated by color change. This approach has been used with many plants: tea leaves, coffee, citrus fruits, neem leaves, and even spices. For example, using green tea leaves extract to synthesize gold or silver nanoparticles is already well documented. There are numerous advantages. So, first of all, the plant extract synthesis is extremely simple and fast. It's economical since plant materials are cheap and widely available. It's environmentally friendly. Moreover, the plant extract often imbues the nanoparticles with a biofunctional surface. For example, nanoparticles synthesized using a neem leaf extract might carry some of neem's bioactive compounds on their surface, potentially giving the nanoparticle additional antimicrobial or therapeutic properties. Indeed, studies have found that the green synthesized silver nanoparticle sometimes show enhanced biomedical activities compared to those made by conventional methods, likely due to the phytochemical capping. Plant compounds can also make the particles more biocompatible. A capping of glucose or polyphenol is certainly less toxic than, say, a layer of CTAB surfactant for the chemical synthesis. On this slide, you can find the summarization of the comparison of methods of the nanoparticle synthesis. You can see the information about size and shape control typical for each of the synthesis method, the scalability and cost, equipment complexity, and eco-friendliness for the physical, chemical, and biological methods of the nanoparticle synthesis. So I hope that this information was interesting for you, and thank you for your attention.